How was everybody's weekend? Good? Mine was great. Well, last week when I missed you guys, I went to see my brother because he got me tickets to that concert and the Train concert with REO Speedwagon. So we were up in Colorado, up in Denver, Colorado Springs, and we were like teenagers again, going to concerts like 3 nights in a row. It was amazing. I was really tired, but it was fun. I've always wanted to see Train, and I don't know how much longer they'll be touring, so that was his birthday present. And then Gregory Alan Ishakoff was the night before in the Springs, which was pretty incredible. I've never seen either of them. So, and you all seen Ishakoff? I guess he came through Flag, but I missed it. So, all right, nobody else has seen him? Anybody know who he is? Okay, couple? All right. What did you guys do for Labor Day weekend? Huh? Camping? Somebody say camping? Sweet. Was the weather nice? Pretty nice? No rain? You didn't get rained out camping? All right. Everybody connected? I know there's a couple of you that we've talked about GRL, textbook stuff, and linking. Anybody else having some issues? Sounds like we might have some issues of them reaching back out to you after you file a ticket or call them and leave a voicemail. So I've looked at the grade book. Most everybody's linking up, and your grades are imported over in a Canvas. So if you're taking it in GRL and it's not importing to Canvas, then we have an issue. We should probably get in front of it early, like this week. That would be great. A couple of announcements. So tomorrow is the last day to add or drop classes without any penalties. So if we're full, this class is full, but if you're contemplating moving your schedule around, I mean, if you know friends that are wanting to get in, they're kind of hovering, waiting for a seat. But tomorrow's the last day. I just want to make sure you guys knew that. And otherwise, there's extra fees associated with it. The university kind of does that to discourage it. You have quizzes 2 and 3 due on Friday. Did you all see that? Quiz 2 and 3 due on Friday. You can do them early if you want to get it in front of them, but they're due at 11:59 on Friday evening. We're going to be talking cell biology today, and last week we had kind of the molecules of life that you watched online, and that was the big Klein Library lecture hall from the spring. That's when we recorded that one. So that's why the room looks different. Their screens are a little nicer than ours here in bio, but I still think the recording's kind of working out okay. And then today we're going to talk about cell biology, and that is really all of the review material to get us all calibrated. So here's the thing is, many of you have taken biology in high school, or you tested out of it. How many took AP Bio and tested out of biology in college? Okay. Some of you had bio a while ago. Here's the challenge that I told some of your colleagues. Hey, it's been a while since I've had biology. I didn't take it in college because I took it in high school. Either I took a co-convened class or joint registration or whatever they call that now, dual enrollment at the community college and in high school, or you took the AP exam. So here's the challenge. If it's the AP exam, they really teach the class in high school to the exam. They don't necessarily teach it for the material that you're going to need in college as upper division or in grad school. Like I'm not trying to knock your high school teachers, but they have to be subject matter experts to be able to teach AP classes. So they typically have to have a master's or higher to teach that class. But they may not have a master's in that particular discipline. And they may not be, they're school teachers, they're not necessarily nurses or physicians, right? I mean, again, I'm not like trying to knock them, just saying they don't know the pathway that you're on and how far you're going to actually have to use it. So there may be things that we're talking about that are relatively new to you, even though you already had the prereq. Does that make sense? So when I talk about, hey, you already should have had this, and you're like, I never had that, that's where that gap is. Again, I'm not trying to throw anybody in the bus. I'm just trying to explain it wasn't that you got a bad education. It's not that you had bad teachers. There's a gap. There's a little bit of a gap, and what we're trying to do is fill that gap. So today is about cell biology, and you're like, what in the world does cell biology have to do with anatomy and physiology? Why would it even matter? I'm going to pose the question to you all. Why would it even matter? Cell bio. Cellular biology. It's the building blocks of us. Every tissue in the body is made up of cells. Okay? Anything else? What else? Why is it relevant? Different shapes of cells, or the morphology of the cell, will lend itself to a different functionality. That's absolutely true. You're going to see that next week in lab, and you're going to see that next week here in lecture. We're going to go through histology and tissues, and you're going to start seeing the way the cell is structured dictates the way it performs. Hair cells, skin cells, muscle cells. They're all cells, but they're all different. Nervous tissue, nerves are neuronal cells. They look very different than a skin cell, but they're all cells. They all have very similar building blocks. But that's why it's relevant. So now if you're treating a disease and it attacks the cell and it attacks a certain organelle in the cell, like the mitochondria, what do you think might be a manifestation of that disease at the tissue level? What happens if the mitochondria are zapped in a tissue bed? Just brainstorm with me. Think outside the box.
There's no energy production. They're not going to be
There's
going to die.
Perfect. No energy production, no ATP being produced. The cell's going to die. You may have necrotic tissue. You may have a loss of function. If it's a muscle cell, it won't contract anymore. If it's a respiratory muscle cell, you can't breathe. That's actually important. Right? So what I'm trying to convince you is that this kind of stuff is actually important and relevant. It's just when you learned it in high school, your teachers may not have had the vision because they hadn't had the experience in college and the university, and because they're teaching high school. And so it's harder for them to tell you why you would need this if you want to be a physician or a nurse or a researcher or a pathologist or an epidemiologist or a PA or a PT or an OT or dental hygiene, nurse practitioner, the list goes on. So understanding how it works, and I love that statement earlier here about the leads to the function. That's absolutely true. And so we're kind of doing our own thing right now in lecture. This is the first week in lab. It's a lot of syllabus stuff, right? Meet your TA, make sure you show up, reserve your spot, don't let them drop you. There'll be a lot of shuffling because tomorrow's the drop deadline, right? Or the change deadline, whatever you call it. Next week, we kind of get on track where they start in lab. We start in lab kind of where we are here in lecture and in tissues and histology. And you'll start to see some of these things where hopefully you can look at it through the lens of, wow, these things look weird. Well, there must be a reason that they look weird, right? There is an intent in the way it was shaped or the way it was designed or organized. It serves a purpose. It's not just by accident that it's shaped that way, right? So you'll see like the intestinal epithelium looks different than the skin. You'll see that skeletal muscle looks different than cardiac, looks different than smooth, right? But at the end of the day, those building blocks we got to cover here, okay? So next week, lab and lecture are pretty much in sync, and we're kind of prepping to get to that space. Quizzes 2 and 3 due Friday. Our change deadline, no fees, is tomorrow. Now, 3 really important questions for you. What does a nosy pepper do? What does a nosy pepper do? It gets all up in your business. No? Okay. No, not good? Okay. What do you call a cat that likes lemons? Cat that likes lemons. A sourpuss. Nicely done. What kind of bird can lift heavy objects? A crane. Nicely done. You're starting to warm up. Okay. So a couple of things. If you ask a question, I'll probably repeat it, so it's on the video recording. So it's not that I'm mocking you. I don't want you to feel that way. I could hand around the mic, or I guess I can throw this box. This box is supposed to be like a tossable speaker, or microphone rather. But I worry I'm going to hit somebody and it's not going to be soft. So I'll probably just repeat your question. The other order of business is the jokes. They don't get any better. You just get a little bit more dull, okay, throughout the semester, just so you know. So what is in the upper left of the screen? Your upper left. What's there? What's that a cartoon picture of? Who is my graphic artist that wants to draw science? Over here? Are you here today? That's awkward if you're not here today. Sorry about that. Okay. Do you guys remember that? That was this class, right? Yeah, okay. So there was a person over here when we were first together, which was like a week and a half ago, right? And they were going to be their interest was in graphic design, but in science. So they're going to make pictures like these. Actually, this is not a drawing. This is an actual data collection. What is the drawing in the upper left? It's a cell. It's not a plant cell, but it is a cell. So nucleus, everybody see the nucleus, the big sphere? You've got mitochondria, these like pod-looking structures. You can see vesicles. You can probably make out the Golgi apparatus and other organelles. This is a schematic of a human cell. It's a eukaryotic cell, that's right. It's a eukaryotic cell, very good. Remember we talked about prokaryotes and eukaryotes in basic bio. So we pretty much ignore prokaryotes until we deal with like diseases, like bacteria, bugs, and things that make us sick. All right? In the lower right, what's this a picture of? What's that? It's not muscle, but why would you say that it's muscle? The striations look like muscle. That's actually a good guess. So what we're staining here are alpha-catenin inside of HeLa cells. So these are the intracellular structure that holds the cell together. But you see that in skeletal muscle that gives it striations of patterns, right? But it's going to be a little different. They're more linear when you see them next week. And we'll show some pictures here in lecture as well. These are actually HeLa cells. These are human cervical cancer cells, affectionately referred to as HeLa cells. Anybody know where the HeLa cell name comes from? No? Henrietta Lacks. Who was Henrietta Lacks? Any guesses? Henrietta Lacks was a woman back in the 50s who passed away in her early 30s, and she had cervical cancer. And her physician got a sample of her tissue and was a prominent cancer cell biologist, researcher, and started growing them in the lab and discovered that they never died. They were immortal. They would divide forever and never give up. And he passed around the cells to his other scientist friends, because that's what scientists do. They're weird like that. We're weird like that. Did not have permission, but you didn't need permission in the 1950s. Well, HeLa cells now are one of the most widely researched cancer cell lines ever. And the Lacks family got how much money for them? 0. So it's been this huge controversy. And this is a website from Johns Hopkins honoring Henrietta Lacks. And you can read about this little story. But that was a picture of the HeLa cell line. There's a great book, if you're interested in reading a book, called The Immortal Life of Henrietta Lacks. And it's quite fascinating. I remember you and I had this conversation. And so this is my tribute to the family and to the patient, educating all of you. We don't always get it right. But when we get it wrong, hopefully we recognize it, even if it's years later, and we try to do the right thing. And so that's what Johns Hopkins is doing, and many, many others. You can see that she died at the age of 31 in 1951, and her cells live on today. And those cells have contributed to cancer cell research for decades, leading to all sorts of different types of therapies. So I ask the question again, why do we care about cell biology in an anatomy and physiology class? Well, if you can understand these building blocks like the gentleman in the back said, you understand foundationally how the cell operates, you understand the basic principles of chemistry and biology like what we lectured on last week, then you can start putting together the storyline of how we're put together, and you have to study how we're made in order to figure out when things aren't working correctly, how to fix it. It gives you a tremendous amount of insight. All right, so that's my long-winded introduction for today to get us started on this path of cell biology. These are the building blocks of our body, cells. And our understanding of cells really began when we could start seeing them. You know, it's amazing what we have at our fingertips. And you've heard old people like me gripe about what it used to be like when we were your age, right? But we're talking about 1600s is when the microscope, in the mid to late part of the 1600s, when the microscope was actually invented and widely used. And all of a sudden, we're looking under this microscope, seeing cork was the first thing that we looked at, cork cells. And we had 200 years of cell observations leading to what we call cell theory. And the microscope was invented by this Robert Hooke, but there were lots of people other than Hooke that Virchow, Schwann, Leeuwenhoek, all of these individuals who studied cells that actually contributed to the cell theory. And so this cell theory took about 200 years to kind of solidify, and there's 4 principles in the cell theory. Anytime you see a list like this, it's a good thing to probably study because it really makes a nice multiple choice question, right? That's like a huge hint if you want to like rewind the video and rewatch that part, okay? So number 1, cells are the basic structural and functional unit of life, and they share certain generalized features. We kind of talked about that. A muscle cell versus a nerve cell versus a skin cell. They all have very similar features. They're not identical, but they have some certain generalized features, like a nucleus, ribosomes, mitochondria, Golgi apparatus, all of the cellular machinery that you've previously been exposed to. If it's been a while, per the introduction, you can review that a little bit. And chapter 3 in your textbook does a really nice job of giving you review. So if this is rough for you, I would really encourage you to read chapter 3. It does a really nice job of explaining cell biology. Organisms are made up of more than one cell. Number 3, cell functions are carried out by subcellular structures known as organelles. We've already talked about some, like mitochondria, the Golgi apparatus, ribosomes. And then all cells arose from pre-existing cells. Cells beget cells. Cells make other cells. Cells divide and replicate. This picture is a picture of the cytoskeleton of the cells labeled with a fluorescent microscope. So the cytoskeleton is the inside of your attic. You go up into the attic and there's all those trusses that keep the roof up. That's what the cytoskeleton does inside of a cell. Now, different than our roof, the cytoskeleton of cells actually can shrink and grow. And that's actually how you get locomotion or cellular movement. That's one way to get cellular movement, is you stretch and then you shrink. You stretch and then you shrink, right? So the cytoskeleton actually has to be dynamic. So like if your house could do that with its studs in its wall and its attic trusses, it would move its position in the neighborhood, right? To the end of the cul-de-sac or, a better spot, which would be kind of cool. What's typical? What's typical? And I kind of use in quotes like typical. Like we draw these typical cells in textbooks. And next week, you're going to see really weird cells. You're going to be drawing cells in lab. You're going to be looking at the microscope. We'll show some of the histology in this class, kind of lines up with lab. But you'll probably not see anything that looks like, well, you'll see something that looks like this, but it's like one example, with like this border that has these villi or microvilli. You'll see that in the intestine. But we have a plasma or a cell membrane. It's the outer boundary. It separates the outside from the inside. The cell interior is the cytoplasm. And remember, the cytoplasm is uniquely different than cytosol, because cytosol is just the fluid inside the cell. And the cytoplasm is the fluid with all of the organelles. You can look at Chapter 3 for a walkthrough of all of those types of organelles. We're going to go quickly through them. But the analogy that I'm going to use is like a manufacturing line. Has anybody ever had a job like in a manufacturing line? Even if it was like in fast food or the restaurant business? Okay? So you know that you have kind of a specific task. If you're the cook or the chef, you make it. You set it on a plate, there's someone that preps it. There's someone that dresses it up, and then there's someone that takes it to the table. There's someone that serves the table. There's somebody that actually clears the table, and then there's somebody that runs your credit card, right? Depending on how sophisticated the restaurant is, you may have like 5 or 6 different individuals in that factory line all to give you a meal. It's not any different if you order something from Amazon, right? It's not any different if you work in textile industry. The cell is like a manufacturing facility. And the director is the DNA. They have the instructions of what we're doing. This assembly line analogy is this endomembrane transport system. The Golgi apparatus that packages things and puts things in vesicles to move them throughout the cell or to have them leave the cell. You have support features like organelles and other cytosol components, like the ribosomes. And some of the products stay inside, like the cytoskeleton. You manufacture protein that's actin. You don't leave the cell with it. You put it inside the cell to create those cytoskeletal structures that we were looking at. But some, like collagen, we're going to look at the skin here in a couple of weeks. You'll look at the integument or the skin next week in lab. You'll be looking at the different layers. Those products that go outside the cell, like collagen, form the architecture of skin. And most of the body's extracellular matrix is made up of collagen. There's about 26 different types of collagen, but for our purposes, we're going to say collagen is collagen. It's made inside the cell, it's packaged in a vesicle, and then it is exocytosed outside of the cell to where it's needed. You do that with elastin. Those are the 2 main extracellular matrix proteins in our body, collagen and elastin. One is stiff, that's collagen, and one is stretchy, that's elastin, like a rubber band. And, some tissues are stretchier than others, like your skin's pretty stretchy. Your aorta is very stretchy. Your bladder is really stretchy. Your bones, believe it or not, they flex, but not as much as skin, but that's why they're not brittle unless you have a brittle bone disease. And guess what? In those circumstances, do you think there's a defect in elastin production? Yes. So there's already a medical example or a clinical example of why we care on how these things are actually made. Because if this system is compromised with collagen production or elastin production, it leads to different types of disease states. A brittle bone disease or a collagen deficiency in certain cases leads to what's called Marfan syndrome. You can Google Marfan syndrome. Okay? That's way beyond the scope of this class, like testing on specific diseases. I teach a general pathology class. We get into some of that stuff later, but dropping some of where we're going to be in the future or where you might go in the future, I think helps you see the relevance to why we even study this stuff right now. So why is a cell small? Why is a cell small? What thoughts do you have on why we have small cells in the body? Like, we're 10 to 15 microns. A frog egg is about 1 to 3 millimeters, right? The largest cell, the giant squid, is 39 feet long. Why did we go from metric to English? I don't know. It's just, right? 39 feet. What is that, like, 10 meters? Ish. An ostrich egg, it's one cell. It's 6 inches long. So it's like 18 centimeters. Why aren't our cells any bigger? Any guesses?
We have a myriad of different cells that all do different things, so we need them to be small. We have hundreds of processes going on.
Yeah. I like that. We have a myriad of things happening. That's a nice word. Myriad of things happening all at once. Different types of cells doing different things. So having them small and compact is helpful. The smaller the what? The smaller, maybe the more efficient. Yep. So you have easier transportation. We're going to look at active and passive transport, which is really important. Over here against the wall. Surface area to volume ratio is a tighter. It's not big volume, small surface area. It's actually big surface area, small volume. Anything new, the gentleman behind you? Everybody already take them? Okay. These are great answers, right? But, you see cartoons where it's like, monsters versus aliens. It's like, the slime guy is just one giant blob. Like, we're not that way. He digests a ham by just, like, sticking it in his... Do you guys know what I'm talking about? Like, when you have kids, and my kids are older now, but when they were young, that's all we would watch, are these, like, Disney movies. So they're small, so we need things to see them. That's why we need a microscope. We can either shine light or we can shine electrons. So visible light's what you're going to play with in lab this week and next week. Probably more next week. You'll get the scopes out. You'll look at slide preps. You're shining white light, visible, full spectrum light at it. If you need higher resolution, you get into shining electrons. And you hit an electron at a surface, you coat the surface, the electrons bounce off the surface, you collect them on detectors, and then you reconstruct the image based upon the electrons bouncing off of the structure. And that's basically electron microscopy. We see it where the light shines off of it, bounces into our eyes, and our retina processes it in the brain, and you see a picture. We can't see electrons, so we need equipment to be able to create an image. Okay. I want to move to this plasma membrane, and before I talk about it, I want you to see a video picture of what this looks like, because I think there's so many misunderstandings of a membrane. Like, a membrane is this, hard, solid, creates a barrier. You can't get anything through it, right? Oh, it's like saran wrap, right? You cover something with saran wrap, right? No, this thing is very flexible. In fact, you can put things right through it. You could poke your finger through the membrane. So imagine like a swimming pool. Anybody grow up with a swimming pool? Imagine you have like styrofoam balls about this size, maybe 4 inches in diameter, and you cover the entire surface of the swimming pool with white styrofoam balls. And then you grab a couple of yellow ones and you throw them into the pool, and a couple of red ones, maybe a couple of green ones, and then you turn on the pool filter, you know how it swirls the water around, and those styrofoam balls all can mix up. That's more of this fluid mosaic model. That's what that terminology means. So I want you to kind of see this video.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move.
It makes that funny sound too when you do it. I'm just kidding.
And begin to regroove. The lipid molecules of the membrane naturally assemble in a double layer because their tails
Bless you.
Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all.
Okay. Beautiful music, too. Is that a little bit more helpful for you to visualize what a plasma membrane actually looks like? Because we like to show these pictures in textbooks. You saw them in high school. You probably saw them freshman year of college. You may have seen it on the AP exam. Right? You'll see it in your textbook. But they're stationary. They're pictures. And you can't really appreciate, that's why I like the videos, you can't really appreciate that this is a moving structure. But it has a hydrophilic and a hydrophobic region. So if you want to put something all the way through it, right, it has a fat or a lipid. So water isn't going to pass through it readily. That's why it keeps the outside stuff outside and the inside stuff inside. But the functions are to create a barrier, to be a gatekeeper, to decide what comes across and what doesn't. It has a support structure at some level. You can actually anchor, if you see down here, you can see the cytoskeleton can anchor to some of these proteins. So there is some structure capability on the inside where you can tether things to the membrane. And it allows for communication from one cell to another, where you can link up one protein from one cell to a protein from the other cell. And the example that I use is in cardiac muscle. There's proteins known as connexins, kind of a clever name, right? Connexin. And they connect one cardiac cell to the other. So when you want to initiate a contraction of your heart, you actually do it in sequence, and all the cells are talking. It's like if everybody lined up in this classroom and we all held hands, and then I had somebody initiate squeezing their hand, and then you squeeze that hand, and then it goes down the line. And do you understand the analogy? So you want the propagation of that signal to occur very systematically, and that's why you need cells to talk. Here's another list of like 4 or 5 things that are functions of the plasma membrane, right? Another really easy test type of question. What's it made of? Well, this is the phospholipid bilayer. There's an electron microscopy picture on the right side of this slide. You're right. And it's demonstrating what this phospholipid bilayer looks like. And then here it is blown up in cartoon format. So your colleague that's the artist that wants to draw science pictures, this is what I'm talking about. All of these drawings in your textbook are made by somebody. Right? It's the artist's interpretation of what these things look like. It's made up of this phospholipid bilayer. It has a polar head, polar phosphate groups, and nonpolar tails, or neutral acids. So they self-assemble, they self-arrange. And that's that fluid mosaic theory or model that we've all grown up learning about. Now, every now and then you'll have cholesterol that will initiate inside or stick inside of the membrane for a support structure. You've probably heard, hey, cholesterol levels are high, cholesterol must be bad. That is not actually true. Excess cholesterol is bad, but you need cholesterol to build a cell membrane. The plasma membrane needs cholesterol. Here's the thing is, you can manufacture all the cholesterol you need yourself. You don't need an exogenous source. You don't need it in your food or your diet. That's why we have an issue. Because your body, if you're eating a well-balanced diet, your body will manufacture cholesterol as much as it needs to repair and to grow. What can enter the plasma membrane or this phospholipid bilayer? It's selectively permeable. Selectively permeable. It allows certain things through, other things it keeps from passing. So small, uncharged, nonpolar molecules will cross easily. Small, non-charged or uncharged, nonpolar molecules will cross easily. Like oxygen, carbon dioxide, urea, that also crosses easily. Water crosses through channels. Any other molecules need some sort of mechanism to get across. So let's talk a little bit more about these membrane proteins because they're the key for getting things across. So you can see right here, this is a transmembrane protein, transmembrane because it crosses the membrane, like transatlantic across the Atlantic, transcontinental across the continent. Transmembrane protein, this spans the entire membrane and has a channel down the middle that allows things to come through it. This is really important. We're going to come back to this in the semester when we talk about muscle contraction, when we talk about nerve action potential, because you've got to actually move ions that have charges through these channels in order to contract a muscle or to fire a nerve. So that's why we talk about these building blocks here, is so when we talk about muscle contraction and nerve action potential, you're not like, oh, it's just magic. I don't know how it works. Okay? All sorts of sophisticated transmembrane proteins. They're embedded in the membrane. They may have a cytoskeletal connection to somewhere else in the cell. And they could have peripheral proteins associated with it, but they're not associated with the plasma membrane. A lot of variations. So what do these membranes proteins do? Well, there's a whole list here. Here's another list, right? For exam 1, unit 1, we've got a lot of list of stuff going on as we talk about these review things. Same kind of principles that we talked about in molecules of life with all the chemistry. Right, when we talked about simple sugars and complex sugars, right? We talked about monosaccharides and disaccharides and polysaccharides. You remember that? We talked about proteins, right, and amino acids. Here we're talking about these proteins and the function of the full protein, transportation, moving thing from one side to the other, recognition. There's a very complex protein structure known as the major histocompatibility complex, MHC. Anybody know what the MHC really does? It helps cells identify other cells in the body. That's correct. The only word that I'd add to there is it helps you identify your own cells. So every cell in your body that's nucleated has an MHC on the surface that's like a name tag. And it says, hello, my name is. And so anything that comes into your body that's foreign, that doesn't have that name tag, that's how you know to kill it and not kill your own cells. Well, what about autoimmune diseases? Right? Well, that's when this self-identification becomes problematic, and that's where we get a lot of these autoimmune disorders. And we talk about a lot of that in pathology, but that's the recognition. That tells you your cell is your cell. It belongs to John or Julie or Sam, right? It's your cell. This is Rob's cell. Enzymes. They catalyze reactions on occasion. They help with cell-to-cell linkage. Cells connecting to another cell. They serve as receptors. They serve as a cytoskeletal attachment point for the inside of the cell. Lots of different functions of these membrane proteins. Most of them are what we call glycoproteins. So they are sugar-coated, right? Like a nice cereal that your mom never let you have. But I'm not bitter about it, right? Every now and then we get like Honey Nut Cheerios, but for the most part, we weren't allowed to have any sugar cereals. Glycocalyx, a sugar coating on the protein. Okay. Different classes of these proteins. So we like to organize them in different classes. We can talk about fibrous or structural proteins that are like long, rigid, rod-like. Collagen is operating in this category. Gives structure inside and out of the cell. Collagen is an example that we talked about outside the cell. You can see it right here. And then we've talked about actin being inside the cell. How do you remember that? Actin, in. Actin, inside the cell. Collagen is outside the cell. If you remember that actin is inside the cell, collagen is the opposite, right? So the structural protein inside the cell is actin. The structural protein outside the cell is collagen. These proteins are not very water soluble. That means they don't go into solution. They don't dissolve in an aqueous environment. That's important, right? Because we're like 70% water. So you don't want us just dissolving all of our structural proteins. We could have a globular protein. The globular protein is a functional protein. It has a purpose other than just structure. So the soluble part is on the inside. It has the polar groups that are out and the non-polar groups in. It has compact globular structures or bunches, kind of like a grape bundle. It's a globular structure rather than a rod. You understand the difference? And so in these globular structures, you actually find an active site. And so this is the molecule hemoglobin. Hemoglobin is a really important protein that is found in our bloodstream. What does it do? What's that? It carries oxygen. Specifically, oxygen has a higher affinity for iron. Right? You know that iron oxidizes quite easily and turns red. So iron and oxygen like each other. So the body has 4 binding sites that are made up of an iron molecule in each of the globular structures of the hemoglobin molecule. So how many oxygen molecules will one hemoglobin molecule carry? 4. Absolutely correct. So when you're measuring the patient's oxygen saturation and it says 99% or near 100% on the pulse oximeter on their finger, what is actually happening? How many, on average, molecules of oxygen are bound on a hemoglobin molecule? 4. When the patient's at 50% saturation, what's happened? 2, and they're probably near dead, right? 75%, there's 3. They're probably on supplemental oxygen, and you're getting really worried if you're in the 70s. That's what that number means, okay? It's not just some magic digital display. You actually are getting a reading or a readout. Think about all of the research that went into building that pulse oximeter so you can just look at the number and go, she's okay. He's doing fine, right? Okay, so this cell protein factory, I would like you, for homework, to put these in order as they go through protein synthesis. If you're confused, go to chapter 3. Put them in order. The starting point is going to be where? The nucleus. Okay? Questions? Just want to pause for a second. So that's your homework. We'll talk about it on Monday. We can also talk about it, you can talk about it in SI sessions and TA sessions. So tomorrow's going to have 7 to 8. 7 to 8 p.m. tomorrow is a TA review session. You can bring questions. You could talk about this. We're waiting on a room. Rooms are a premium right now. So we're in queue for a room. If we don't get one for this week, she's going to send out an announcement. It'll be on Zoom, tomorrow's review session. And then Eliana's, I gotta go to my text here. She told me hers, and she posted these on Canvas. Tuesday, 4 to 5 p.m. in the Science Annex Room, 117. Wednesdays, 11 a.m. to 12 p.m., Science Annex Room, 122. And Thursdays, 5 to 6 p.m. in the Science Annex Room, 122. And those are already in Canvas. So Tuesday, 4 to 5, Science Annex 117. Wednesday, 11 to 12, Science Annex 122. Thursday, 5 to 6, Science Annex 122. And then Amara's will come out here soon if we can get another room, or it'll be, worst case scenario, by Zoom. Okay? Lots of resources, like 4 different slots each week. And in my office hours, you guys remember my office hours are Monday and Tuesdays, 2 to 3. Okay. So let's talk about what happens inside the cell. We talked about the mitochondria. We talked about a poison that kills the mitochondria, renders it useless. A mitochondrial toxin would actually suffocate the cell because they wouldn't be able to manufacture ATP. And I say suffocate because really oxygen is necessary in the mitochondria for aerobic respiration, which is where you get most of your ATP from. But glycolysis, remember glycolysis, we net how many ATP from glycolysis? 2. Somebody was like flashing me gang signs, but you're correct. That's 2. Well, they kind of have the 2 sideways, so I was like, okay. 2, net ATP from glycolysis. So where does glycolysis take place? Where does it happen? In the cytosol. If you said cytoplasm versus cytosol, I think you're splitting hairs there. I think you could argue it happens inside the cell, but not inside the mitochondria. So it's in the liquid part, which is the cytosol. Cytoplasm is kind of the same thing, but including organelles. So I like cytosol best, but just kind of food for thought. Some of these things are kind of arguable, right? What about the Krebs cycle? Where does the Krebs cycle occur? Mitochondrial matrix. Absolutely correct. And then the big one is the electron transport chain. Where does that take place? In the inner membrane of the mitochondria. And the amount of energy that you generate goes from top to bottom, least to greatest. Now, am I super excited about you memorizing how many comes from each? Not necessarily, but depending upon how you count, if you take one full run through aerobic respirations, you'll get somewhere between 36 to 38 ATP. You really only can count one way on glycolysis. You take one cycle through glycolysis, you net 2. So it's 2 versus 36 to 38. And the big difference is oxygen, in the presence of oxygen with the electron transport chain, you generate significant amounts of energy. Why is energy so important for the cell? And we're going to talk about this. And you're going to start seeing, I think, more insight as to how some of the cell biology really impacts anatomy and physiology function. But here's another way to look at this. You've got proteins on the upper left, carbohydrates down the center in green, and fats on the far right. All 3 of those fuel sources will actually go into glycolysis and generate ATP. You can see this, indicator of ATP. Well, you can take an intermediate, pyruvate, from glycolysis and feed that into the Krebs cycle or the citric acid cycle or the tricarboxylic acid cycle. It's got 3 names for the same damn thing. Sorry about that, but that's the way it is. So let me say that again. The citric acid cycle, the Krebs cycle, and the tricarboxylic acid cycle are all the same thing. That's the middle wheel. Maybe that cleared up like years of confusion for you in general biology. You're like, I thought they were different things. They're all the same thing. They were just different investigators that were studying it, calling it different things. Like Krebs, Dr. Krebs called it my cycle, Krebs, right? It's like the Keller cycle because I'm studying it. Then the electron transport chain just has one name, and glycolysis just has one name. You can further move into the electron transport chain in the presence of oxygen and generate significant amounts of more ATP. So let's talk about some terms I want you to define. You could write these down. Let's define glycolysis right now. What is glycolysis? What is it? Glycolysis. Break the word down. Glycolysis. Splitting of a glucose molecule. Well done. To generate ATP. Is glucose a complex sugar or a simple sugar? Is glucose a complex sugar or a simple sugar? Simple sugar. Is it a monosaccharide, a disaccharide, or a polysaccharide? It's a monosaccharide, which is the simplest sugar you can have. Remember, your body wants it in a simple format. So if you have a polysaccharide, you're going to break. All it is like take like a gold chain. All those little links on a gold chain, each one of them is a glucose molecule. So you have 2 links, that's a disaccharide, 2. You have 1 link, it's a monosaccharide, that's 1. That was last week's lecture. You have many, many, many chains on your gold chain, links of glucose, that's a many saccharide. Saccharide is another name for sugar. I told you, this is almost like a foreign language, right? Monosaccharide, disaccharide, polysaccharide. You have simple, complex sugars based upon the Latin root word. Glycogenolysis. Glycogenolysis. What is that word? Turning glycogen into glucose. Glycogen, is that a simple sugar or a complex sugar? It's complex. Is it a monosaccharide or a polysaccharide? It's a polysaccharide. So glycogen is an example of that gold chain necklace. And then when you start clipping the links, you make individual glucose molecules. And that's called glycogen lysis or glycogenolysis. I call it glycogen lysis so you can see the word in there. Gluconeogenesis. This is like 3 words run together. Gluconeogenesis. What is that? Let me say it this way and then you go, okay? Glucose, new, beginning. That's actually literally what the word is. Glucose, new beginning. So what is it? Okay, perfect. Because if you break down the word, it makes sense, right? Glucose, new beginning. It's the production of new sugar, new glucose, using a metabolic pathway. You can make it from scratch. And then glycogen. We kind of already covered glycogen, but what is glycogen? What's another name for glycogen? How do you define it? It's a polysaccharide of glucose molecules. It's the body's favorite way to store glucose. So if you have extra glucose after a meal, right? How many were athletes in high school? Raise your hand. Okay. How many of you are athletes now? It doesn't have to be like you play for a professional team or, an NAU team. You could do intramurals or you compete competitively. How many of you did like carbo loading before an event? Okay. How many have heard of it? Okay. So the night before a big event, race, match, meet, competition, whatever it is, you have a meal full of lots of carbohydrates like pasta, and there's a lot of freed up glucose that at evening your body stores as glycogen that you have available for the next day. That is actually a real thing, and it actually, there's a lot of science to it, the carbo loading. Okay, we did that together. So there's really no sharing comparing, like we did it together. You perform a chemical analysis of a bone called an osteocyte. Osteo is the root for, or the prefix for bone. Cyte is the suffix for cell, bone cell, osteocyte. You find it contains large amounts of pyruvate. You determine that what has occurred? Glycogenolysis? Okay, we got C and we got E. Why do you say E? Who said E? Why is it E?
Because probably the other license would
Glycolysis? Yes. Like, glycolysis is likely the source of energy, and if it's glycolysis to make pyruvate, that's way up here. The other lysis, I like that. That's okay. That's how you remember it, right? You're breaking down words. It's like when you learn Spanish or you learn French or German or Chinese or whatever it is, Mandarin, whatever it is that you learned, you had little tricks and tips that you had to figure out what is it. This is a foreign language, folks. And the sooner you kind of practice with it, and that's why we have 4 review sessions a week. I have 2 office hours. There's 100 of you and there's 3 of us, right? That's 1 per every 33. It's a pretty good ratio, okay? All right, we're shifting gears into passive membrane transport. So for like the next 19 minutes, we're going to be talking about moving things across this membrane. Because that's really the goal, is to separate outside from inside, allow certain things to come in. And so now you'll start to see, like, okay, I think there'll be some examples, maybe some light bulb moments where you're like, oh, actually, that kind of makes sense now. It costs energy to do that, but you burn energy here so you can go do that and move molecules in that direction. Oh, that's kind of clever. Yeah, there's a lot of cleverness here. So membrane transport. You probably recall somewhere in your science education that the term diffusion has a specific meaning. Who can share with us what diffusion means? What's the definition of diffusion? If you're struggling, it's actually on the board. Okay. Down a gradient. So from high to low, right? Follows the law of entropy, right? Of thermodynamics. So you move from a high concentration to a low concentration. That's the term of diffusion. So we also know that a solvent and a solute equals a solution. Like, how many of you wear contacts? You have saline solution on your countertop. It's water and sodium chloride. And it's called a solution because it's a solvent, water, which is the best solvent, and the solute, sodium chloride, NaCl, and that makes your solution known as saline solution for your contact lenses. In cells, the extracellular matrix is never equal to the the extracellular environment is never equal to the intracellular environment. Do you see that little equation there? This is stating a truth. In cells, the extracellular environment is never equal to the intracellular environment unless the cell's dead. If the cell is dead, then there's no homeostasis. There's no difference between the outside or the inside. In fact, if you poke a hole in the cell, it's a great way to kill it. And a lot of pharmaceutical strategies to kill, like bacteria, are to actually ruin the membrane and lyse the bacterium. Okay? So the extracellular environment is never the same as the intracellular environment. We're always moving stuff. If we're always moving stuff across the membrane, do we need to make energy all the time? Yeah, nothing's for free, folks. You cannot move an ion without paying energy to make it happen. When you were a kid and your mom or your dad or your guardian said, go clean your room, you're like, right? Because you know it takes energy. It doesn't just automatically go from disorder to order, right? You can't just snap your fingers and it's cleaned up. Like, you have to put the energy in. So to create a concentration gradient that's useful, it takes energy to do so. That's the fundamental building block like your colleague so eloquently said in the beginning, that's really important to appreciate. I mean, otherwise you just memorize and go, I don't know why we do this. We just have to create these gradients or whatever, you know. But if you can kind of get behind like, okay, the how and the why behind it, it becomes quite interesting. So we have a couple of different ways to move things across the cell. Why do we have to move things across the cell? Well, remember an action potential for a nerve to fire, a muscle contraction for you to breathe, or your heart to beat? You need to move these ions for those things to happen. If these ions don't move, you're dead. So your patient's not alive anymore. So it's really important that these things are always moving. And that's what we spend the bulk of our energy doing. That's where we spend the bulk of our ATP. There is some passive transport that can take place, and we'll explain how that happens. No energy is needed. It just moves from high to low. Active transport, energy is required. You move from low to high. You're setting up the gradient. Like, for example, how many of you come from like a rural town? Perfect. How many have driven across a rural area? How many have seen a water tower, like in the Midwest? And you're like, why do they store their water way up there? Like, that doesn't make any sense. Just so they can put their name on it? Why do they store their water in the middle of America way up high on a tower? Why is that?
For water pressure?
Water pressure. Potential energy becomes kinetic energy. You go from a high concentration to a low concentration. So we do this as humans, right? That's the whole concept of a water tower. Sugars, ions, and amino acids move either passively or actively depending upon this concentration gradient. And you can kind of see a picture here of how this plays out, where here's a membrane. This is ECF, that's the extracellular fluid. ICF, the intracellular fluid. Here's a membrane. Here are particles that are in high concentration. You can see there's only 2 of them here. This is just a generic molecule. Later, we'll start labeling like sodium and potassium. We'll label them sugar or glucose, a simple sugar. Right now, just for the illustration point, you're seeing that they're going to move from high here to low. And now instead of 2, there's 3. And how do you get it in? You got to go through a transport molecule, a protein, a transmembrane protein that regulates when it comes in, how much comes in, how much energy it takes. So simple diffusion. Simple diffusion for lipid-soluble substances is very, very easy. Simple diffusion for lipid-soluble substances means that like things like oxygen, carbon dioxide, and fat-soluble vitamins, you don't have to spend energy to get them across the cell membrane. Oxygen, carbon dioxide, and fat-soluble vitamins. It's like, well, that's a weird one to throw in there. Fat-soluble vitamins are the ones that are very dangerous and you can overdose on. Water-soluble vitamins are fine. You can have as much as you want. You just excrete it in your urine. So you have to be cautious about having too many vitamins that are fat-soluble vitamins because you could actually build toxic levels inside the cell. There's no regulation. Oxygen and carbon dioxide, really important for the cell to give off carbon dioxide as a waste product and for it to bring oxygen in as a nutrition. This figure that's here on your right is showing osmosis as a special case of diffusion. It's actually involving water. So osmosis is a principle of diffusion, but it's following the water, not the solute. So if the solute cannot move and is an area of high concentration, then the water will move to the area of high concentration solute. So water, the terminology we use is water will follow the solute. So water will move into the area that has high solute concentration. I'm going to give you a clinical example here in a minute of why this is so important to understand. You can actually kill somebody by giving them a bag of water instead of a bag of saline in their IV. And it's because of this. Okay, you can rupture their red blood cells. And unfortunately, this has happened in clinical medicine more times than we would like to admit. So this red blood cell is abnormal. It shouldn't look like this. I just put this question here before I teach it to you to engage the audience if you guys already understand this. Do you think this was in an isotonic solution, a hypertonic solution, or a hypotonic solution? If you don't know the answer, that's fine. I'm going to teach it to you here in a second. But for those who have already tracked and kind of pushing themselves a little bit, which solution was this red blood cell put in? Which one? Hypo? Hyper? How many say hyper? How many say hypos? Oh, it's about even. It is a hypertonic solution. So the solution is too high in solute, and so the water is leaving the cell, and the cell is actually shrinking. It's actually crenating. So let me explain it in this way. That was just kind of a challenge question, but now I want to actually explain it to you evenly, okay? Actually teach it to you. So the middle picture is what a red blood cell should look like, right? It's biconcave, so it's like a disc with a donut hole that doesn't go all the way through. It has like a dimple on both sides. That increases surface area. That allows for the oxygen to readily come out of the red blood cell and has a higher surface area to do so by having it biconcave versus just flat. Kind of a clever strategy. Isotonic solution, equally balanced, is the middle picture. Water is going to move down its concentration gradient in these situations until it reaches an equilibrium. That's what goes in that blank. On the left picture, your left, that is a picture of a hypotonic solution. Hypotonic solution. So the bottom one. Hypo is this one. Iso is this one. Hypotonic means it's got too little solute, the solution on the outside of the cell. So if it has too little solute and the cell has more solute than the outside, water is going to rush into the red blood cell and it's going to rupture. So if you give a bag of water to a patient, you're bathing the red blood cells in water. The water is going to rush into the red blood cell and lyse the red blood cell, and your patient's going to die. So you have to hang a bag of saline, normal saline that's isotonic. So by extension, the far right one is a hypertonic solution where you have too much solute outside, the water leaves the red blood cell, and it crenates. It kind of like shrinks. Make sense? Okay. So let's look at this in a slightly different way. Let's look at it with this picture question. You've got each of these, you're going to take a 10% glucose, a cell that has 10% glucose inside of it. You're sticking it in an environment that's 20% glucose, distilled water, which is basically just water itself. Like, students are like, what's distilled water? I was like, have you never bought distilled water at the grocery store? But okay. This is just regular like water. You buy at the grocery store, distilled water. Sometimes like steamers take distilled water, or your iron is supposed to still, because there's no like solutes in it. It doesn't have any calcium, so it doesn't mess up your iron. Does that make sense? You're like, oh, that's why Mom always yelled at me for not using distilled water. And then here's 10% glucose. So 20% here, 10% here. So I want you to say in each situation what's happening. Is there no net change? Is the cell swelling? Is the cell shrinking? I'll give you a second to answer these questions. Okay, question 1. What happens? Right there. Question 1. The cell shrinks. Cell shrinks in question 1. Right? Because where is the water going to go from the cell? Out into the beaker. Because the beaker has more sugar. The beaker has more solute, so the water is leaving the cell, so the cell is going to shrink. What about question 2? Any takers? Now everyone's trigger shy. In the back.
The cell swells.
The cell does swell. Yeah. It's okay. It's okay. It's not very specific, but you're right. Cell swells. I think Emirates was going to pick the same thing. Okay, what about the last one? Equilibrium, no net change. Okay? Questions over that? That's a great one to review in SI section, TA sessions, to kind of grasp, okay, what's happening here? Ions are moving. All right, we're going to finish it out here in the last 5 minutes with some facilitated passive diffusion. So these are things that like transport bigger molecules, like amino acids and sugar. They're protein-assisted diffusion. We call them channels or pores. Most of them are gated. You can either regulate them using electricity, that would be voltage regulated, or you can regulate them using a ligand, like you actually have to have something bind to it. A ligand would be like a